everyone. We're going to go ahead and get started. Thank you all for coming out for this year's ROCI Research Day. We are going to go ahead and get started with our kickoff lecture. And we want to give a big thank you to Dr. Josh Brown for coming to visit us all the way from Florida. I'm going to try to read through his very impressive bio for you guys, but he'll have to correct me if I mess anything up. Josh Brown is a PharmD PhD who graduated from the UK College of Pharmacy PhD program in December 2016 and accepted a 10-year faculty position at the University of Florida College of Pharmacy. He's focused on real-world evidence for medication safety and effectiveness, and his research has been funded by the Food and Drug Administration, National Institute on Aging, the Bill and Melinda Gates Foundation, and the state of Florida. As a PI, his total funding includes $2.6 million in the last 5 years. He has contributed to projects with over $10 million in funding, and his publication record includes 130 published works to date, with 91 of these being first author or lead author, and 23 trainees featured as first authors as well. He has mentored and sat as a chair for many PhD and pharmacy students throughout his time, and we are so excited to have him here today to present to us. So I will give it over to you.
Sorry, that's big hands and little mic. All right. So I'm really excited to be here. I've told anyone that'll listen, this is my first time being back since October of 2016. So last time I was in a room in this college, I was defending my dissertation. So really excited to be back and kind of get to present some of the work that I've been doing and continue to do, but also I think a little bit of an information lecture will be appropriate here. You'll get to look at real world evidence, which I think will be important because we've probably got a pretty diverse crowd here. So you'll see that I've got a little two-part title here, and I'll explain a little bit more of that. But I'm going to be talking to you today about real world evidence, real world data, so RWE, RWD. The whole field that I think generally I'm focused in, and several of our own colleagues, like the folks in POP, do similar work. And the first part of that is in quotes, because that is a real thing that I've been asked by a reviewer before. Isn't everything in the real world? So let's go through some stages where we'll actually define and talk about what real world evidence is before we get into what I've actually done with it. So we'll define it. And then I'm actually going to go really quickly, I hope, through 3 different case studies where I can show you how real-world evidence can basically be used to development, to do academic research, and really lead to some meaningful, clinically important evidence that can impact patients' lives. So for anybody else that doesn't know me, I'll provide quick introductions as well to myself. So I was born and raised in Arkansas, so did undergrad pharmacy school in Arkansas. And I came here as the Humana Pfizer Fellow. Technically, I'll call myself the first and the last of that because I did by Dr. Talbert. It's more complicated than that, but it was a really unique chance for me to get exposed to managed care organizations, pharmaceutical industry, and so on, and also get my PhD at the same time. And since then, I've been down in Florida, working, I feel like, a lot, and producing quite a bit of work in addition to my teaching and other roles. So I'm going to give you a weird disclosure slide. So again, everyone that'll listen, I tell them I have been on parental leave the last 2 months. So we've got a 2-month-old daughter at home. And my disclosure here is if I stumble through something or this entire presentation doesn't make sense, it is her fault, okay? And as someone asked me earlier today, well, does that mean you finally get to get a good night's sleep? I thought that was going to be the case until the building outside my hotel room here in downtown Lexington caught fire last night. So we had a 5-engine fire situation right outside my window. So no sleep, no sleep. All right. So what? So I wanted to find to understand the context of term in general of how people like myself might have used it in the past, but now that it actually has an official definition, it's important. So when I was in grad school, that was 2014 through 2016, evidence studies. So it was really just kind of a coined term that we use to differentiate ourselves from purely like epidemiological studies, which they just call population level studies, and to really differentiate RCT, which again kind of came in the title of, well, isn't even an RCT in the real world? And we'll get to that point. But this question, the came, and I don't know if any of y'all have been through peer reviews, but the eponymous reviewer number 2, whether or not they're actually reviewer number 2, sometimes they're 3 and so on, is just that person that's never going to love your work, right? So this person asked me this question. I submitted as a grad student here at Kentucky. And so I really was silly because we got rejected anyway. So reviewer number 2, what is it actually officially? So I used to use that term kind of loosely for everything official definition that I think is really important for you all to be aware of. So with the 21st Century Cures Act, it's one of the last bills that President Obama signed before he left office. It actually, the FDA man program and to incorporate it into our drug approval process. So there are lots of kind of history components to it. So proponents thought it would help drug development, speed it along, provide new evidence for better treatments for the market. Of course, there are opponents out there that, of course, hate anything real-world evidence or epidemiological in general because of issues like confounding, and RCTs are our gold standard, right? But the 21st century said, we got to do it. So what did they come up with? So FDA was basically required to accelerate this drug development process. They had to produce guidance on what that means. And they specifically pointed out certain uses of real-world evidence. And that included things like supporting approval for new indications for products, or simply using real-world evidence to evaluate safety. And we'll talk a bit of why these are important, how they can be used as we move forward. But we do need to define real-world data again, and this all comes directly from the FDA guidance. So it's no longer like a coined little term that I use to describe all of my research, but FDA first defined real-world data. And that is data relating to patient health status or the delivery of healthcare that is routinely collected from a variety of sources. And this is any data coming outside of a clinical trial. So this can be things like administrative claims data, EHRs, patient registries, really just anything. It could be wearables. So if you take data from my Apple Watch, that could be considered real-world data. And then real-world evidence is basically evidence generated from that real-world data. So it's, if you use real-world data to generate evidence, you've created RWE. And again, this RWE versus RCT is like, I feel like I'm in this eternal battle of arguing this over and over. It's basically part of my job at this point. But RCTs being our gold standard for drug approval and obviously for evidence in general, I think it has very clear benefits, obviously randomization, and certain measurements of outcomes that might be difficult in other settings and so on. But there are a lot of other key differences. So it's experimental, certainly, compared to real-world evidence, which is almost always observational. So we're not doing experiments. We're not determining who gets treatment or not. The patient populations, RCTs narrow them down very specifically, and I'll provide a very kind of striking example of why that might be important. Whereas real-world evidence, it is from the real world. So we're taking it from the full set of patients or the population that might be using the product. RCTs are short-term, super expensive, right? So drugs can be approved with 2-week RCTs or 1-year RCTs or 2-year RCTs. Whereas once a drug is approved, how long is its life cycle? Potentially forever, unless it's pulled from the market at some point. So we have the potential to capture things that are long-term. And then obviously the cost of real-world evidence is probably one reason why it was a clear option to incorporate into the regulatory process because it is relatively cheap. So I apologize for some spacing, it's smushing together for some reason. But I like to point out when I talk about the difference between RCTs and real world evidence, right? We'll talk about how that looks in a little bit. Their interactions with physicians are different. The cost is free. The drug is free. Their adherence is almost usually perfect because it's supported by a lot of interactions with the clinical trialists. The healthcare system is different because technically you're not even inside the US healthcare system if that's where you're doing the study. And oftentimes even the comparison that they're conducting is not even meaningful. So from my anticoagulant research days, one example I like to use is like the pivotal RCTs for Pradaxa or dabigatran. Adherence to dabigatran in the real world at 6 months is like 50%. I've done those studies myself. In the clinical trials, it's like 95%. Patients are taking it constantly, twice a day, every day on schedule. The comparison was to warfarin, which at that time was meaningful, but clinically meaningful now. No, we want versus Eliquis versus Xarelto versus whatever other DOAC might be now recommended by the clinical practice guidelines. So RCTs can't really keep up with this changing paradigm. And I like to teach with memes. And so I ask my pharmacy students every year to create some memes after I teach them some of this difference in RCTs and RWE. So I'll present some of that up there. So I can't take credit for it, except for being the inspiration, of course. Apologize to the other faculty out there, but enjoy some of these. We'll talk about this in a specific example. So I think a lot of RCTs and even the FDA approval process oftentimes just looks for that nice p-value, which no one really knows what a p-value is and what it can be used for. So statistical significance versus clinical significance will be an important topic here. And then my students think this is funny. And my biggest issue with RCTs is really generalizability, or that's in another term is the explainability. So how applicable is an RCT to the real world population that will be treated? And we'll go through a specific example here. And I want to bring up RCTs as a gold standard. I'm going to point out a recent approval where even the drug approval process, I feel like, failed us. Did the RCT tell us much? So this is an example of Aduhelm. I don't stick with the product name, so we'll just call it by its brand name, which is the first-in-class monoclonal antibody for beta amyloid for treatment of Alzheimer's disease, right? Anybody pay attention to this one? A bit controversial, so it's a really interesting case example. So guess what? It works really well for its mechanism of action. You can basically see in that plot, I know it's small, but you can just see the precipitous drop in beta amyloid. That's what we're measuring there versus placebo. Great. We also had some really interesting and serious safety issues. So microhemorrhages in 40% of patients, and a lot of those were clinically significant safety concerns. The interesting part of that as a biomarker is that it's also, at that time, was not considered a recognized surrogate endpoint, but it is a surrogate endpoint that was measured in clinical trials. But they also measured patient-reported outcomes. And I'll point to two of these because these patient-reported outcomes are measured on a certain scale. And we like to point out things like a minimal clinically important difference, which is how big of a difference has to happen for this actually to be meaningful. So for these two that I point out, I just pulled these values that come from the literature and other studies. And you can basically see the difference from placebo is less than 1, right? Whereas the minimum difference that would actually be meaningful to patients and in a clinical setting is at least 1 point or up to 2 or 3 points. So this drug hit its biomarker, hit a surrogate endpoint, doesn't produce patient-reported outcomes. And obviously I kind of buried the lead there, but in the end it was approved. And why does this matter? So one, the surrogate endpoint component not being an accepted surrogate endpoint was one part of it. But then the generalizability issue that I pointed at is maybe, to me, the most important component, especially for someone that's done a lot of older adult research and looked at drug safety in older adults. So this was a study published in one of the JAMA journals. And these people actually just took the Aduhelm RCT that led to its approval and applied its inclusion-exclusion criteria, particularly the exclusion criteria. And you can see how the specific exclusion criteria kind of play out. But what they basically conclude is that 90% of the patients that are going to be eligible for the drug, so 90% of the patients that have Alzheimer's disease or mild cognitive impairment, would have been excluded from the clinical trial. The other parts that this doesn't necessarily include is that it's also not representative of the demographics. So off the top of my head, about 16% of the population is Black. I think 30% of patients with Alzheimer's disease are Black. And if I remember right, it was something like 1% of the RCT population studied was Black. So there are also disparities introduced by RCTs that again make a case for real world evidence being quite important. So where did that leave us for that drug in particular? Well, that's a drug that's probably going to be now investigated using real world evidence. So they received approval based off of that surrogate endpoint. They have to do some follow-up studies. And then you might have paid attention to Medicare Part D. I'm sorry, it's not Part D, it's actually Part B, is going to cover the drug, but coverage with what's called coverage for evidence development. So it's basically going to be a pragmatic clinical trial in a real world population. And we're funding it as taxpayers instead of Biogen. And they're going to collect all this clinical data from real world data resources. And then just as a side note, Medicaid has to cover it because they have to cover at least one drug per American class, but they might put other exclusions in there. I also want to add one more example because it's a case where formatting really jumped, where in that case I feel like the RCT went awry, and now real world evidence is going to have to step in and fill in. This is actually a case where part of the Cures Act now allows companies to submit real world evidence with their initial approval documents. So for Karyopharm Pharmaceuticals, they have a product that was approved for multiple myeloma eventually called selinexor. And they actually presented their clinical trial results and real world evidence. And we'll talk about why this is important for the role of real world evidence. But essentially, once you get into penta-refractory multiple myeloma, your patient size is really small. So real world evidence is an idea where you could supplement your clinical trial with an external comparison. But they screwed up. So if you actually look at it, the real world evidence is the first column, the RCT is the second column. So I'm telling you that usually the real world evidence group is going to be worse off than the RCT group, which is why we need real world evidence to study these drugs long term. But in this case, I don't think they paid enough attention to that, or they didn't have a good epidemiologist on their team, where they actually presented real world evidence that made their drug look a lot worse than even the clinical trial. And that's because they introduced a bias. It's not too important, but they introduced mortal time bias. So basically, to be included in the clinical trial, you had to survive a certain amount of time to get treated, and they didn't do the same in the real world evidence group. So in the end, the FDA completely rejected their real world evidence. So this is a case where it went bad. All right, so some of the roles for real world evidence specifically. So the first of that is related to primary approval. So real world evidence actually has a role in the initial approval. So a new drug that's never been approved before. And this is particularly important for any like rare diseases. So if you're looking at an orphan disease where there might be 100 patients, and this really occurs in the US that have a condition, you can't necessarily get enough people to randomize. So you might be able to recruit 20, you provide them treatment, and you don't have a placebo arm, right? Or maybe it's not even ethical to treat them with a placebo. So instead of actually treating someone with placebo, you might have collected the real world evidence on the same patients before they were treated, or you might take just real world patients from another data source that have the same disease, but they're not part of your clinical trial. And you run basically a parallel comparison. So that might be like an external control arm, sometimes what that's called. So that's one way that it can be, real world evidence can be involved in the initial approval, or you could use it to get a second indication. So once a product is out on the market, real world evidence or real world data is being generated constantly through the pharmacy claims data, through the EHR and so on. So particularly for things that might have an off-label indication, you could see where they might officially get maybe an actual indication for that if patients are using it for that purpose. You could also see it in situations like with the Aduhelm example and really many, many others, where that product is involved with some kind of accelerated approval pathway, where maybe they only have to have one clinical trial, maybe that approval is based off of the surrogate, and now we can use real world evidence to do long-term follow-up, maybe of the same RCT population, or maybe just people receiving the treatment in the real world, and then evaluate whether or not it gets a full approval for the actual indication that it's treating using real world evidence. And then for me, the most clear application is for safety. Clinical trials are short and they're small. relative to the population that's going to be treated. So being able to look at long-term safety issues in big populations is the whole field of pharmacoepidemiology, which I've called myself a pharmacoepidemiologist at some point, so it's important. And the last bit that I want to introduce about real-world evidence, it had that whole regulatory framework, but I also want to point out for anyone that might interact with real-world evidence in the future, there are so many groups that are using it for decision-making, particularly payers and pharmaceutical companies for various reasons, including talking to each other using real-world evidence. And then it's also, I think, important to realize that it also has a role in clinical practice. So this might be real-world evidence as I used to think of it. So it still has that role, but now it definitely has a regulatory role that's very clear. And the other thing that we have to deal with is now that we have real-world evidence, the evidence generation process has to be much more high quality. So they're talking about things like registering observational studies. So I've never registered one of my studies, but now pharmaceutical companies do that so that they're not accused of like cherry picking their results. And we know when studies happen, so it holds them to a higher standard, similar to like clinicaltrials.gov, right? So we're starting to emulate that process. But it's also important to realize that there are high stake situations and low stake situations. So if you're going to get a new indication, that's pretty high stakes. If you're just trying to compare it to an existing product, maybe that's a little lower stakes. And if it's really changing how patients might use a medication in the real world, it's definitely higher stakes overall. So I'm now going to switch to my own real world evidence studies. I'll actually pause just for fun and see if anybody wants to clarify any questions at this stage before I talk about my own one. Yeah. Alongside it. So they do it in a few different ways. A lot of times it might be an external control arm, like I was describing, where there are established patient registries for certain diseases, particularly rare and orphan diseases. So you might actually use that registry to recruit people into your RCT, and the other people that aren't recruited in, you just continue to follow them prospectively, because that's basically a data source you might have access to. You can also collect it within like a pragmatic clinical trial. So that's a situation where oftentimes they might randomize clinics to certain interventions. For example, it exists within the healthcare system as it is. So you are collecting, you're basically your outcomes, all the measurements that you're including might actually just come from the EHR or from the clinical assessments that occur. So that's a place where real world data is technically being collected, although you're still within randomization framework. Great question. All right. So I promised somebody a discussion about generic drugs. So that's our case study number 1. And this is one of the first projects I got to be involved with early on. So we basically sat down with the FDA and the Office of Generic Drugs, the Office of Pharmaceutical Quality. And I was lucky enough to be in the room, probably inappropriate given the topic they were talking about, which is mostly PK/PD modeling or pharmacometrics, and that's what a lot of my collaborators do here. But what they were mainly interested in is, could they develop some PK/PD models for specific drugs to understand any formulation variability and how that could lead to bio-inequivalence of generic products. So, this is an issue where, of course, the FDA requires PK data to establish bioequivalence for a generic product. Then you have a generic product, and we might get 13, 14, 15, and maybe even more of them out on the market made by as many different companies. So we assume that it leads to the same physiological response, but of course, to expedite the availability of generic drugs, we do not require really any outcome collection. So the issue here is imperfect manufacturing compliance, and poor quality is certainly still possible. And I'll put a book up here in a second as I animate through this slide, but a recent book called Bottle of Lies tried to shed some light in a bit of a sensationalized manner about this imperfect system. So in general, a lot of manufacturing, of course, is moved overseas where the quality controls cannot be monitored as effectively. So different excipients used, different particle size, things like that could lead to a generic. Probably a lot of pharmacists and others might have heard some of the cases where ADHD meds not working as well, Synthroid not working as well when people switch to generic, and there are a few other examples. So the FDA surveillance on this, they incorporate a lot of different components to use what's called a site selection model, which is how they decide who they're going to monitor and inspect. So that includes the site characteristics, so the specific manufacturing site. They include things like their compliance and inspection history. So if you had rats in your hopper at some point, you're probably going on a bad list and will be inspected again. The product characteristics, so more complex formulations, like maybe extended release products, injectables, anything that might not be oral. Those are more complex products to replicate as a generic. And then this idea of hazard signals. And what they typically have used in the past is real world data, but typically what a system called FAIRS, which is basically a spontaneous adverse event reporting system. So it's really passive surveillance. FDA is sitting back and waiting on somebody to tell them there's an issue. So as I'm sitting in that room, I generally ask the question of like, is there a potential role here for other real world evidence? FAIRS or AIRS counts as that. The facility reports counts as that. Recalls count as that. But the real world evidence that I was specifically pointing them towards was a system called the Fda Sentinel System, which is basically a big data system of real world data that the FDA has created as part of one of their other mandates. But this specific office had never used it. So I said, well, let me take over and we'll see if we can use basically similar system like the Fda Sentinel System to evaluate generic issues. And that's the book I was pointing to. A bit sensationalized, but it's worth a read for anyone that's interested in being scared of the medications you take. So we did take a step back. So essentially started a new collaboration. And this is a place where if you're ever doing any translational science, where you're crossing the aisle to folks from pharmaceutical sciences, drug development, to maybe the pop type groups, we really had to figure out what each other was talking about. And that is quite a time investment. I think it probably took 2 years to really get the terminology down. But we really came up with a framework, and it was important to define this, particularly as we want to write more grants or present why this collaboration is important and show the role of real world evidence. And we came up with 2 main systems of knowledge here. One was the idea of orthogonal hypotheses, which basically you're pursuing the same hypothesis, you're asking the same question, but you're doing it in very different ways. They're doing PK/PD modeling. I'm doing real world evidence generation using epi, or just a general knowledge transfer signal detection idea. So I observe something in real world data and I go, can you figure out why this happens? Or they know a lot more about pharmacology than I do. I've slept a bit since pharmacy school, so they can actually inform my study designs and so on. And I have some pretty good examples of that as we move forward. And then our specific jump into this research project, we decided to pick a model drug. And in this case, we picked All XL. When it went generic, it had some almost immediate issues with some of the generics that came out from specific manufacturers. So this timeline is a little bit hard to read, but as early as, I believe, 2008, there started being recalls of a Nesher product, and I believe Activist made a product that was recalled. But even as recently as 2018, the same product has had issues with formulation variability and basically failure in its bioequivalence, and has been recalled multiple times over this period of a decade. So what my colleagues did in the PK/PD part, don't ask this slide, I understand some of it, but they basically wanted to look at the PK profile in these situations where the formulation parameters are changed. So they included the brand, some generic versions that had known issues, and basically looked at bioequivalence, which is what VE stands for, looking at the area under the curve, as well as Cmax. And you can tell in a few places that relative to the brand product, the test case is falling outside of our typical bioequivalence measures. So they established that the formulation parameters appear to be important, but what they don't capture with PK is what? The outcomes. And still the outcomes are arguably most important. So they did inform a bit of my real world evidence studies here though, and I should mention that based off of some of their observations, we decided to incorporate outcomes related to either subtherapeutic doses or particularly supratherapeutic doses. So you can see particularly that an issue with these products was dose dumping. So people would take something that's extended release but a good portion of that drug would come out immediately. So we could predict adverse effects. So that's a safety issue. But you can also see in some cases, it seems like the drug might wean off a bit earlier than predicted. So you might have issues related to it not being able to treat the indications that it was there. So I won't spend too much time on this. This is mostly for my other epidemiologists, pharmacoepi folks. But we essentially did what I said. We basically wanted to compare that time period in 2007, 2008, when these generics first came. So we have a brand product that's on the market, and we have generics available on the market. And we wanted to compare people that initiated them and see if they have any of these safety or efficacy concerns within administrative claims data. So again, very similar to this Fda Sentinel System. We defined a few different outcomes. Again, safety and effectiveness, I think, were obvious and very important. But we also realized that for some drugs that might be really hard to measure, especially if you want to even do high throughput screening. So we came up with what might be considered a universal outcome of discontinuation or switching. So patients are our best measure of whether or not a drug might be different. And we hear this all the time, right? Patients have complained for years about certain generic medications. Could be a nocebo effect, so it might be a little bit in their head, so to speak, but it also could be real. So when they detect that, do they stop taking their medication, or do they specifically switch back to the brand product? And then in any pharmacoepi study, we always want to have a good comparator. So in this case, we had a good active comparator that we identified, where there was a generic formulation of amlodipine benazepril that was approved on the same day, which is also for similar indications, cardiovascular indications, and is similar, usually once daily dosing. So it provided us a relative comparison where we can now compare generic metoprolol ER versus brand metoprolol XL. At the same time, we were doing kind of an indirect comparison of, for a product that doesn't have known issues, this amlodipine benazepril, do we see similar issues which might be more related to that nocebo effect? Running through this quickly, what we essentially saw is that with the amlodipine benazepril comparator, we see no signal of a safety issue or lack of efficacy issue. But for the metoprolol succinate generics, we see, and this is just within the first 3 months of use, almost an immediate effect on ER visits. I put up here just heart attacks as an outcome. So we see heart attack rates increase by over twice as much. We saw similar effects on angina, on arrhythmias, on hypotension, and other outcomes that we measured, similar kind of 2 to 3 times increased rates of those. And so basically we chalked that up to being able to identify these generics that have issues, and basically told the FDA, we were able to do this within 3 months of a generic coming to the market. So we think that you could use your Fda Sentinel system to evaluate every new generic drug actively. So instead of sitting back and waiting 2 years for an article to be published in the New York Times about all these individual patient experiences, or you taking 2 or more years to collect enough FAERS data for you to do something, you could use this system to identify all these generics that have had issues. And the FDA stopped talking to us, and even pulled names off of the publication that came out of it, although this was a FDA funded contract, because that would basically jeopardize potentially a lot of the generic drug industry, and basically would say that their systems in place weren't doing a good enough job. I do know that they've internalized the system, so they hired ORISE postdocs and some other scientists to do similar work, and they put out some more recent work looking at specific drugs like Synthroid generics and using other real-world data resources. So generally kind of interesting for me there. Within the same project, looking at generic drugs, we also were interested in kind of a more recent example, and in this case, it's more of a prospective example. So we wanted to look at anticoagulants, and we specifically looked at dabigatran in particular because, again, we're looking at potential new generics that might have formulation issues or have other characteristics that make them a high-risk product. So dabigatran being a prodrug, having low bioavailability and some other formulation parameters identified by my PK folks, we decided it's a good target drug. And then what's shown on the screen is basically its PK/PD profile for both stroke and bleed risk. So the middle dots, those that are actually closest to each other, are basically the reference points. So this is what we would expect for annual stroke and bleed risk if a generic was bioequivalent. And what my PK/PD friends did was modeled out what an 80% bioequivalent generic might look like. So that's our lower end. So you see that as you decrease the bioequivalent parameter there to 80%, which is like our threshold for approval of a generic product, bleed risk goes down fairly quickly and stroke risk increases, but not all that much. And you can see some key differences between the stroke and bleed risk curves here. And then as you get to that higher level of 25% higher bioequivalence, that's what our kind of rightmost dots are. Those are your new bleed and stroke risk if you're more exposed to the generic product. Built a pharmacoeconomic model. I'm not going to spend much time on this. So a lot of this is generated using RCT data. We did throw in some parameters generated from real-world evidence and created a very detailed and robust pharmacoeconomic model. So this is basically a simulation of outcomes and even costs in this case, because we wanted to know not only what were the clinical outcomes, but is a generic product always cost effective compared to the brand if it's not as safe or as effective. So we basically looked at those extreme scenarios where you have a upward bound, 25% higher exposure generic dabigatran or a 20% lower generic dabigatran. And quickly go through this. What we basically saw is that your lower bound is always going to be cost effective. And you can see that a lot of these results are actually driven by bleed risk, which makes a lot of sense if you remember that very steep curve related to bleed risk. So the lower bound's always better. You minimize bleed risk. Your on-treatment stroke is always pretty low and not too sensitive to drug exposure. But what was kind of interesting for us from the kind of economic standpoint is that compared to the higher bound generic, which has, compared to brand, has over 2,000 more bleeding events predicted, and very few strokes are saved from that, the brand product would actually beat out the generic product. And that was very sensitive to the generic cost. But I think particularly early on, generic products aren't as cheap as we think they are, especially with initial approval and prior to actually having multiple multi-source generic access. So some lessons learned here for the specific products. Again, lower bioavailability is kind of preferable. But again, talking to the FDA about how this type of modeling could be used, incorporating clinical outcome modeling as well as PK/PD modeling. We thought it really gave them an idea of being able to model this before generics come out for specific products and understanding the directionality of how they might want to reset some standards. So for anticoagulants, where that bleed risk increases really quickly and stroke risk does not really improve much as you get higher and higher exposures, we basically thought, and I think they generally agree, that they could start setting different thresholds, particularly for like anticoagulants, to make sure that the bioequivalence might be pushing more to the lower end if it ever was a concern. I also like to mention that this is a situation that I actually got to use the same economic model with a pharmaceutical company. And I introduced this particularly for our students here that might not know that these types of decisions are made, but only 5% of products that are developed are actually eventually approved by the FDA, right? So there are failures at preclinical, phase 1, phase 2, phase 3 stages. And companies use similar models, the parameters of their products that are coming out, to basically decide if something is worth marketing. So I got to use the same model for a pharmaceutical company that I can't name, thanks to non-disclosure agreements, but they had what would at that time be a 5th oral anticoagulant hitting the market. So it's really late to market, and it comes down to some of their parameters. Does it have unique pharmacokinetic profile? Like, is it going to be once daily dosing? That would be nice. Nope, twice daily. All right, so you're not going to make a big splash. Is it more safe? Is it more effective? And based off of its affinity to some of the targets, it didn't seem so. And then we run basically some analyses, including that model, what their predicted market share is, and basically at a phase 2A, basically tell them that you probably shouldn't waste your time. And in that case, they actually did drop that product and didn't develop it. So that anticoagulant is sitting in some drug library and may or may not ever be picked up again. And then case number 3 was an interesting example of how to apply real-world evidence here, but again, within the same collaboration. So this is a project funded by the Bill and Melinda Gates Foundation, but it's kind of weird because they're functioning more as the drug developer or sponsor. So what Bill and Melinda Gates wants to do is actually bring a long-acting injectable hormonal contraceptive to sub-Saharan Africa. But they want to get FDA approval for a few reasons. One, FDA approval is kind of the gold standard internationally, so that probably gets them through some hurdles and even distributing in Africa. But they also have expressed interest in selling the product in the US to generate revenue to support that program. So FDA approval is really important to them. But what they're really trying to do, and this is a case where hormonal contraceptives have been available for longer than I've been alive. So there's lots of data out there. So they were really interested in, can we use the existing data to skip or expedite as many development phases as possible in a good way? So they're not trying to cheat the system, but they basically want to say, like, why do we need to do phase 1, phase 2 when someone already did it for at least this molecule, maybe not as a long-acting injectable. So one caveat that was presented to them is that, well, you're trying to put a hormonal contraceptive in Africa where TB still exists, malaria exists, HIV, AIDS exists, and all of these medications in particular that we don't have to be concerned with much here in the US, introduce drug-drug interactions, which might introduce new safety or effectiveness concerns. So our big barrier here in this case, particularly for expediting clinical trials, particularly the drug-drug interaction parts of those clinical trials, was the FDA labeling. And particularly the FDA labeling for hormonal contraceptives says that all hormonal contraceptives are the same, particularly for progestins and for their drug interaction potential. They basically defined it as a class effect, levonorgestrel versus norgestimate versus drospirenone. They're going to have the same outcomes no matter what. And basically we said, we don't think that's true. So this is an idea of what the Gates Foundation is trying to accomplish. They basically want to say these DDI studies that might happen around phase 2, if we can develop enough models, maybe even real world evidence, we can skip some of this and get to approval faster, or at least maybe we can use real world evidence and some of the other modeling approaches to maybe at least make the clinical trials smaller. So expedited, cheaper, in getting a product that has a lot of unmet medical need in the region that they're shooting for. So our friends at Bayer did a study for us where they basically provided the initial data to say that the FDA labeling guidance is not true. So I won't over describe this essentially, but what they basically looked at is in the presence of a strong inducer, so strong CYP3A4 inducer, here's the disposition of all of these different progestin components that can go into a hormonal contraceptive. And what you see is that they're not all equal, right? You can see one group in particular is not all that impacted by this drug interaction. And then there are another cluster that is impacted by this drug interaction. And then again, learning from my pharmacology friends, we start defining the pharmacological parameters that might be important. And these are things that I probably would have been naive to if I was trying to design the real world evidence study without doing the collaboration. So we looked at a few different parameters of importance. Fraction metabolized by CYP3A4, basically saying how much of that product is metabolized by CYP3A4. And the lower essentially is the better here, particularly if we're concerned with CYP3A4 inhibitors or inducers, because that means the drug would not be impacted by that interaction. And another component is sex hormone binding globulin affinity. So this is basically a protein that's floating around. If drug is bound to it, it can't be metabolized. So a higher affinity is better. So what we basically started targeting is maybe levonorgestrel is our preferred product. Maybe norethindrone might be a least preferred product. So we developed this idea, we didn't develop it, but we used an idea called bracketing. So exposure bracketing to say, if that's our best case with levonorgestrel, this is our worst case. Can we again show that within the data that we have? So challenge here, we've established PK/PD. So how do we make a US-based RWD study that supports a study for drugs that are going to go into Africa? And this is essentially where the proof of concept mattered more so than the specific comparisons that we were doing. So we don't use a lot of CYP3A4 inducers. The ones that we really use are mostly our anti-epileptic drugs, either for epilepsy or for other indications. So carbamazepine, oxycarb, topiramate are commonly used, particularly topiramate, because weight loss and migraine indication, off-label uses. So we designed a study around that, and another interesting component here is, our outcome in this case. If you have a CYP3A4 inducer, your drug levels decrease, which means they're not effective. So our outcome in this case is unintended pregnancy, which is not an easy thing to measure in real world data, especially on a population scale, and I can provide more details for anybody interested. We're basically looking at conception as our outcome. And what we're basically able to do is take all of those pharmacological parameters, all of our rank order hypotheses based off of those parameters and the PK studies done, and basically recreate that in a population level database. So we can recreate that rank order, and this is the order of impact of the drug interaction. So in this case, we found like norgestimate was worse off, followed by drospirenone, and then as particularly predicted, levonorgestrel was the least affected by this drug interaction, which led to levonorgestrel being now their candidate progestin product that they're now trying to get in a long-acting injectable formulation. So along with that, as they know that they're going to have to interact with regulators. Again, I said FDA created a real-world evidence definition, but other regulators real-world evidence is still relatively new to them. And even though FDA created a real-world evidence definition for us, there's still only certain groups that actually probably know that definition. So my interactions with the Office of Generic Drugs, they didn't know what real-world evidence was. So we got a chance to show proof and concept of how they could use it for their specific mandate and evaluating generic drug quality. So we got a chance to present all of these findings and the kind of conceptual model of our collaboration to groups like other groups within the FDA, particularly clin pharm groups, EMA, MHRA, so different countries involved, and really started querying them more specifically about real-world evidence and how they see its role, particularly in earlier drug development. So they certainly agreed that they could observe a clear role, and most of them noted that before we even get to use of RWE, they probably need some education about it. So I got to do similar kind of talks to those stakeholders. And of course, they're more used to very controlled data, animal models, RCT data, and so on. So more acknowledgement of strengths and weaknesses, again, more of that education component. And also, we ended up presenting US-based real-world evidence for drug interaction for a product that we're eventually going to push for approval in sub-Saharan Africa. So, the need for developing real-world evidence resources in other countries, which is going to certainly be a challenge. But they also did provide us some interesting feedback on what they view being newly presented with real-world evidence as a concept as potential uses or applications of real-world evidence. So you can see some of these are really interesting, and some of it is what's already defined by the FDA guidance, like the external comparison arms, addition of indications. But they also pointed out use of highly specialized populations. So pediatrics, pregnant women, it's a huge unmet need of evidence that real-world evidence is naturally suited for. Safety surveillance, potential new drug interactions that come about, changes in doses and so on. And there are specific use cases where real-world evidence has been actually used. Can certainly provide some selected readings. Plenty of pubs out there on the topic, but some general concluding thoughts here. So there are very clearly some opportunities to leverage real-world evidence from very early clinical trial phases, so drug development processes. But of course, real-world evidence exists as well within health economics and outcomes research. That's HEOR, more of an industry term, as well as market access. So it has a lot of role, particularly from the regulatory standpoint, and how pharmaceutical companies and maybe payers use them. And it's really growing as a term and has so much buy-in from internal and external stakeholders within FDA, payers, biopharm companies, clinicians, academia, but it has a long way to go before I think it's adopted and perceived more positively. And some of those growing pains include some of those standards and methods that I mentioned. Some of our methods using biostats and epi design are sometimes easy to teach, enough at least to make our students and other people that haven't quite been so experienced be more dangerous than useful in some cases. So there's this idea now of regulatory grade real-world evidence, data standards, methodological standards, transparency and reproducibility, things like protocol registration, and having a similar clinicaltrials.gov even for these real world evidence studies. But again, I think it has a role. So to steal one of my students' memes again in particular, the idea of inclusiveness, of generalizability, of getting evidence that's meaningful to real world populations, not relying on those really curated RCT populations, and then just letting a drug run wild, I think it's certainly got a role there. So I'll acknowledge FDA, Gates Foundation again, specific collaborators. So my PK/PD friends are those within the Center for Pharmacometrics and Systems Pharmacology down in Lake Nona or the Orlando area, particularly Stefan Schmidt, and then our group, Center for Drug Evaluation and Safety. And more baby and cat photos, because I have vowed to never do a presentation without a cat photo, but now there's a baby, so you got to do that. So feel free to email me. I'm sure that was a lot of information quickly, but happy to entertain any questions or discussions with the 7 or so minutes that we do have. And thanks for your attention. Yeah, so that's really what we wanted to follow up with if the FDA would have continued to support our work, but they weren't interested in it. So the way that we were actually going to get around that was interesting is, and first I'll say that that's probably the most important part of it clinically to think about, because patients typically don't have a choice unless they can pay those higher copays, right? So unless you get that DAW put on your prescription, you have to, in most states, dispense the generic. So those substitution laws are in place. So for the patients that can choose, if they're going to pay for it completely out of pocket, they're obviously going to probably be more well off. And you really nailed it. Socioeconomic status certainly does matter for our health outcomes. So the way we were going to work around it, one was to establish the issue of the nocebo effect. So we still wanted to get the patient perception part of it down. So we started designing studies. We didn't do them in the end, where we were going to evaluate not just brand versus generic, but also include the authorized generic. And for those that might not know what the authorized generic is, that is the generic version that is made by the same company. So the pills are identical, come from the same plant, they just have different names attached to them. So they would hypothetically have the same effectiveness and safety unless there was a patient perception issue. But otherwise, the main thing is to then supplement your data set and make sure that you can measure socioeconomic status, as well as base healthcare status. So we can measure their health status prior to initiation. If you're worse off then, you're probably going to be worse off later, but we can use study design techniques or statistics to make sure we are controlling for that or getting it out. But certainly you nailed it. That's a great question. Very important. I didn't look at y'all the whole time. Sorry. It's really about the idea that we don't have randomization, right? So we spend all of our time trying to emulate the clinical trials, and we can't do that because we're not in control of it. We do it statistically or with designs, but I think when I first graduated, I thought I knew how to do real world evidence, and I've learned so much just as like lifelong learning, right? I've learned so much since that I realize now I didn't know what I was doing, 6 years ago, but I feel like I can do it more justice. So there's just not enough people that are well enough trained to think about all of the issues. And it comes down to like the generic issue, like, thinking through like nocebo effects. Like that's a patient perception. issue, right? So how do you incorporate that into study design? Or just generally, like, people in the real world have different factors that prescribers incorporate into their decision making. So in RCT, it's 50-50. Flip a coin, you go one way or the other. In the real world, it might be 70-30 for a given person to receive one treatment versus the other. So getting around those basically selection bias issues and other confounding issues like socioeconomic status is what's really hard. So we have ways to do that. First way that people are really kind of popularizing now is what we call target trial emulation, where instead of doing a real world evidence study that is inclusive and generalizable, you actually design it the clinical trial. So if I wanted to evaluate Aduhelm, I would do all the same inclusion criteria and get it down to almost the same population. And if I can then copycat the clinical trial results, I can then expand out to a more generalizable population and feel a bit better about like my overall design. So that's become one of the more common techniques used is trial emulation. Yeah, so the data that I dream about in my little pharmacoepidemiologist dreams are systems like the VA or Kaiser Permanente, where everything is actually integrated. So you have not only EHR and people probably not even going outside that health system, but you also have the claims because they're also the payer. So those are the things that we are jealous of, and those are very closed systems and hard to access. But my issue with like EMR data is that it's actually not as closed system as those are. So the example that I always use, so that's good for certain things, but like even issues like adherence, like how often are you going to see them, right? So the example that I have there is where so our college is huge on pharmacogenomics, specifically clopidogrel and other antiplatelets. So they wanted to do a real world study about the outcomes occurring, and they wanted to follow up long-term people through the EHR only. And what they basically found was that you don't know what happens until they come back to the system, right? So you don't know if they died. If they died, they're never coming back. So you have to have that EHR system linked to some way to capture death information. You don't know if they're still taking the medication until they come back and you ask them. But you still don't know if they're reporting accurate medication taking behavior unless you have some other supplemental database to fill that in. So they ended up in that study only really having meaningful follow-up of really just the discharge time. So it depends on probably the use case and how frequently you see people and basically how useful that system might be. But in a lot of cases that I've seen, it's better to have both than to say the system captures, like EHR is great for capturing like biomarkers or other clinical measures, and that's really where we use it. So much gets lost in clinical notes though. So obviously like natural language processing and making that data more meaningful is really important. But I still see a lot of value in having like claims linked to EHR, linked to birth and death certificates. Like that's when data really becomes rich to us, because some of that follow-up issue is real big. It's a great question. Yeah, I mean, the FDA certainly is thinking more about how they inspect manufacturing plants and so on. So they recently announced that they no longer are going to announce their inspection, which they have to announce their inspection in China and places where they have to basically be invited, get a visa and so on. So they've announced that somehow they're going to do that. So I think they're continually improving. I think the main thing really is the educational campaign that we kind of followed up with was more focused on clinicians and patients. And I told someone earlier, like clinicians believe your patients, right? As a pharmacy student, I was told generics are all equal to their brand product. Like that's gospel. And apparently it's not, right? Students believe them. And I even had my own mother-in-law. She takes metoprolol ER, got a new version and fainted like the next day. And it's like, oh, okay, well, what generic manufacturer are you using? And can we ask your pharmacy to specifically just stick to that one? And so she ended up switching pharmacies because the other pharmacy still had it. So in that case, I mean, it just becomes an educational component of letting people know that what they're experiencing is real, acknowledging it as clinicians and providing some way for them to get a better medicine, essentially. So I think that's probably most important. All right, we're definitely over time. So I'm happy to answer any other questions offline. But again, I appreciate your attention. Thank you.
Thank you all for coming, and thank you to Dr. Brown again for traveling all this way and giving such a great talk.